Hi guys. Okay, I promised we would go over our assignments, and so here we are. Notice I'm on the enzyme regulation homework page, and I'm just going to talk while we look at the answers. So in this one, it says that examine each substrate and enzyme and color the inhibitors below the enzyme, blue for non-competitive, red for competitive inhibitor. Then answer the questions below. So you see the substrate, which is the star, coming into the active site. And then you see that there's an allosteric site. That's where the inhibitors can attach. And notice that there's some inhibitors down there. The blue ones are non-competitive. The red ones are competitive. The reason the red ones are competitive is because they will attach into the active site instead of the allosteric site. And the blue ones will attach into the allosteric site. So they're not competing for the same site. They have their own special site. But both of those inhibitors, once they attach to the enzyme, they will cause the enzyme to change shape and therefore to not work anymore. So you can see the answers here for both the non-competitive and the competitive for each one, okay? And again, that has to do with the fact that it hooks into the specific sites. All right, going down here, question number 4 is what's the difference between a competitive and a non-competitive inhibitor? How can you tell the difference? Competitive inhibitors compete with the substrate for that active site, and they are often shaped just like that, like the substrate. Number 5, many poisons that affect humans are inhibitors of important enzyme processes. Carbon monoxide is one of these inhibitors. Using the internet, find out what enzyme does it affect and whether it is a competitive inhibitor or a non-competitive inhibitor. So carbon monoxide affects enzymes that use oxygen in their active sites. One of these enzymes is the transport chain protein in aerobic respiration, and it is a competitive inhibitor. Okay, that one required you to do a little bit of research. These do as well. So use the internet to look up the names of each of the known respiration inhibitors, match them to the enzyme that they inhibit. So rotenone inhibits the electron transport chain. Oligomycin inhibits ATP synthase. Alanine inhibits pyruvate kinase. And PEP inhibits phosphofructokinase, which remember we learned about that later. You did this page before we learned about the phosphofructokinase. Okay, number 10, examine a glycolysis diagram and explain why you think that phosphofructokinase is inhibited by high levels of ATP. Why would this be beneficial to the cell? So a cell would want to inhibit phosphofructokinase if there is excess ATP so that it can save its glucose for later. Okay, examine a glycolysis diagram and explain why you think that phosphofructokinase is activated by high levels of AMP. That means low-charged batteries. If there's lots of low-charged batteries, it wants to make more, right? So a cell would want to activate phosphofructokinase if there's excess AMP because there is a lot of AMP. It means the cell has very little ATP. Some glycolysis inhibitors are being studied as anti-cancer treatments. Why? So use the internet to explore this topic and explain your answers. So glycolysis inhibitors are being studied as anti-cancer treatments because scientists have found that cancer cells tend to speed up their respiration reactions. If scientists can slow down glycolysis in cancer cells, they might be able to slow or stop cancer growth, which I think that's pretty cool. Okay, that was your enzyme regulation page. Moving on. Let's do our enzymes and substrates page. This top part was very easy, matching the star to this one, the triangle to this one, the oval to this one. Number 4, many of you did this incorrectly. You drew a reaction, but it wasn't this reaction here. So your reaction should have looked like this with 4 spots for the substrate to come and attach to the active site. It attaches and then it pops off and forms a 3 and a 1, like up here. And then number 5 is There's another way that scientists think enzymes fit specifically with their substrates called the induced fit model. Look up what this means and describe how it is different from the lock and key. Well, we talked about that in class, so you should have been able to refer to your notes and look up the induced fit model. The induced fit model, the enzyme's active site changes shape to fit the substrate as the substrate enters the active site. In the lock and key model, the enzyme's active site does not change shape. Okay, that was the enzymes and substrates page. Carbon cycle. Got some emails there. Carbon cycle, here we go. So carbon cycle, the cellular carbon cycle. In the diagram below, fill in the name of each molecule, organelle, or type of energy. Then answer the questions below. So you see up here, we have a bunch of CO2 molecules. These are CO2s. Then we have a bunch of H2Os here. Here we have solar energy coming in from the sun, affecting the chloroplast. This is the chloroplast. Then we have glucose and oxygen that are formed from this process. They go to the mitochondria. The mitochondria, this is the mitochondria, this whole structure. Energy is stored in the ATP and lost as heat. And this, remember, creates the carbon dioxide and water. So we start with the sun. It goes to the chloroplast and makes glucose and oxygen. It goes to the mitochondria, makes carbon dioxide and water. And you guys now know the entire chemical formula. We've written it several times. We're going to write it down here. So it says, which energetic process takes place in the chloroplast? Photosynthesis. What are the products of that set of reactions? 6 oxygen molecules and glucose are the products. Which energetic process takes place in the mitochondria? What are the products of that set of reactions? So aerobic respiration, we call it cellular respiration, takes place in the mitochondria, and 6 carbon dioxide molecules and 6 water molecules are created. Write the complete balanced chemical reaction for the chloroplast reaction below. So that's the 6 CO2 plus 6 H2O yields 6 O2 plus C6H12O6, which in words, of course, is carbon dioxide plus water yields oxygen plus glucose, but it asks for the balanced chemical reaction. So you need to write out the chemical formulas and them in balance. And you see the coefficients, the 6's that are balancing out that glucose. Okay, number 11 is the opposite, because remember, when we are doing cellular respiration, it is the opposite of photosynthesis. So we have 6 O2 plus C6H12O6 yields 6 CO2 plus 6 H2O. This diagram above shows the source of energy for all life on Earth. What is it? The sun's energy. The diagram does not show the molecule that cells use to store chemical energy, their main battery. What is our battery? Adenosine triphosphate, our ATP. Okay, that is our cellular carbon cycle page. Next. Okay, now we're on respiration and free energy. I know this one was a little more difficult because some of these words are new, but I do expect that you can do some research and find out some of this information. So the diagram shows the overall combustion reaction that burns glucose in the presence of oxygen to form carbon dioxide and water. It also shows the chemical steps respiration uses to break down glucose into carbon dioxide and water in the presence of oxygen. This is in general. So use the diagram to answer the following questions. So this is the breakdown of sugar. So we're starting with glucose in the presence of oxygen. That means it's aerobic, and it breaks down into carbon dioxide and water. So it's showing here, using the respiration enzyme, combustion, and no enzymes. So when combustion happens and there's no enzymes, this is how energy happens, what the energy within the system. And in this one, when you use the reaction enzymes, this is how it changes the reaction progress. So, number 1, what are the reactants for this reaction? Glucose and oxygen. What are the products? Water and carbon dioxide. Write the complete balanced chemical equation below. 6O2 plus C6H12O6 yields 6CO2 plus 6H2O. Is this reaction endergonic or exergonic? You guys have not necessarily heard these words recently. We talked about it back in May when we did physical science, for those of you who had me. Exer means release or energy release. Ender means the energy is kind of absorbed or taken in. So this is an exergonic reaction. How do you know? Because you started out with more energy in the system, and over the course of the reaction, the energy was released. So that's exergonic. Okay? Describe how the course of the reaction is different when the glucose is simply burned or when it is broken down. I just had a Elmo laugh at me. Okay, so when glucose is simply burned, the activation energy is high, and the energy is released in one fast step. Okay, one fast step. So you have to put in a lot of energy here. You have to put in a lot of energy here, and then as the reaction happens, I'm not sure if you can see my cursor or not, but if you could see my cursor, this region right here, oops, yep. So the part where it goes up the steep hill, the solid black line goes up the steep hill and comes back down. This is the activation energy needed. You have to input energy, that's like catalyze the reaction, but not with enzymes, okay? In this case, we have to put in a lot of energy to kind of make the process happen. Whereas with the enzymatic reactions, the activation energy is lowered and the energy is given off in many small steps. Okay? Is delta G. Delta G is the energy of the reaction. Is it the same in the catalyzed and the non-catalyzed reaction? Explain why. So delta G is where we start and where we finish. The difference, remember, delta typically means change, especially in science, it means change in. So we have a change in energy. So we start here and we go down here. It's the same for both reactions. So it should be yes, the delta G is the same because the energy of the products and reactants did not change. Okay? Krebs cycle. So the Krebs cycle. Match each part of the diagram with the correct molecule name or phrase name. Only carbon atoms are shown in this picture. Just slightly simplified. So it asks for pyruvate. Pyruvate is the 3-carbon molecule here. It's outside the mitochondria. Remember, we start in the cytoplasm first for that little precursor step, and that's what gets us going. So pyruvate is letter C. Acetyl-CoA is letter H. Because remember, acetyl-CoA is the combination of the pyruvate and the coenzyme A coming together, acetyl-CoA. Number 3 is oxaloacetate, is letter I. Okay, oxaloacetate is letter I. It's the 4-carbon molecule that comes together with the acetyl-CoA and ends up forming our letter G over here, which will end up being our citrate. So citrate is letter G. Then it asks for mitochondrial outer membrane, that's J, that's the outside membrane. The mitochondrial inner membrane, the mitochondrial inner membrane is letter K. Carbon dioxide are all the single carbons with the 2 white dots on the outside, and here it's letter M. So this is our letter M. It's not showing, in this picture we are not showing the 2 oxygen, so this is just our carbon dioxide. Okay, I know that one might have been a little confusing. No worries. That's why I'm going over it now. Mitochondrial matrix is letter L. That's just the inside of mitochondria, inside the inner membrane. And then the products of the citric acid cycle is everything inside the box in the center, which include the 3 NADH, the 1 GTP, the 1 FADH2, and the 1 and the 2 CO2s. Okay? Great. Now let's answer the question, where in the cell does the citric acid cycle take place? Remember, citric acid cycle and Krebs cycle are the same thing. It takes place inside the mitochondrial membrane or matrix, excuse me. Number 11, what molecule reacts to produce acetyl-CoA and carbon dioxide? Where does it come from? Pyruvate. It starts with pyruvate. And it comes from our first process, glycolysis. What are NADH and FADH2? Why are they important? These are our electron carrier molecules, and they carry those high energy electrons, which when we get to the next phase, which we haven't talked about yet, but we will, the electron transport chain or oxidative phosphorylation, that will be where we see what happens with the electrons. Number 13, fill in the blanks. So we start with acetyl-CoA plus the oxaloacetate, and that yields citrate and coenzyme A. For glycolysis and 2 rounds of pyruvate oxidation and the citric acid cycle. Products of 1 glucose in respiration, and how many of each product is produced. My goodness, so many emails. So, for glycolysis and the 2 rounds of the pyruvate oxidation and the citric acid cycle, how many of each product is produced? So remember, it has to happen twice. We started out with 1 glucose in glycolysis, but it broke down into 2 pyruvate. Those 2 pyruvate both go through the Krebs cycle, and they produce a certain number of each thing. Well, look up here. We ended up with 3 NADH, but this is for 1 pyruvate. So we have 2, so we need 6 NADH. That's why this is here 6. Then we ended up with 2 CO2s, so we need 4 for both of them. We get 2 FADH2s and 2 GTPs. Outside the mitochondrial membrane, we get 2 CO2s and 4 NADH, because up here you see 1 CO2, and I can see why many of you are confused about how many NADHs. If you go back to the fact that in glycolysis we created NADH as well, I think that's where we get 4. Okay? All right. So we got 2 NADH created during glycolysis plus the 2 for each pyruvate, 1 per pyruvate. That is our 4 NADH. Okay, moving on. You did a glycolysis activity. Our glycolysis activity, let's go through this. To answer the questions, how many steps are there in glycolysis? 10. The carbon-carbon bonds are the source of glucose's energy. When you break a carbon-carbon bond, energy is released. How many carbon-carbon bonds are present in glucose? There are 5. Remember, there's 6 carbons, but there's only 5 bonds between the carbons, so there's 5 bonds to break. How are the 2 phases of glycolysis? What are the 2 phases of glycolysis called? Now, we didn't necessarily talk about this in these words, but we kind of mentioned the fact that we have some payoff steps. So you could put preparatory and payoff steps are what we can call that. How many ATPs are used up per glucose in the first phase of glycolysis? We have 2 that are used. Remember, we use 2, but we make 4, but that's a net of 2. So that's the next answer. How many ATPs are built per glucose? That's 4. Subtract the number of ATPs used up from the number of ATPs built, and that is our net gain of 2. What other molecule stores energy harvested from glucose? NADH. What phase is the other energy storing molecule involved in? The payoff stage. What are enzymes that create or break down ATP often called? Remember, kinases are the ones that add those phosphate groups. So the kinase. What are the enzymes that create or break down ATP often called kinases? If you were to come across an enzyme with the word isomerase in its name, what do you think its job might be? Well, we talked about this a bunch. And its isomers are the rearrangement of the same chemical formula, just in a different arrangement. So an isomerase is an enzyme that rearranges the atoms of different substrates. In the 2 pyruvate molecules, how many carbon-carbon bonds are left at the end of glycolysis? Well, you can tell if it's 2 3-carbon molecules, then there's 2 bonds in each, there's 4. Fill in the values of the net reaction of glycolysis below. We have 1 glucose plus 2 ADP plus 2 phosphate groups plus 2 NAD pluses yields 2 pyruvate, 2 ATP, and 2 NADH. Questions on the next page. Glycolysis is an example of an enzymatic pathway. Each product is a reactant for the next step of the pathway, sort of like a cellular assembly line. What do you expect would happen if all of the enzyme enzyme 3, which is the phosphofructokinase, was shut down or stopped functioning? Well, the whole pathway would stop producing pyruvate and ATP, and the whole process after step 3 would halt. So step 3 is the decision point where the cell decides if it's going to carry out aerobic respiration to collect energy from the sugar molecule or not. Cells decide based on whether they have enough ATP or not. If there is a lot of ATP around, the cell will not want to break down more sugar and will want to store the sugar. So ATP is an inhibitor of phosphofructokinase. ATP molecules can stick to the enzyme and allosteric sites and cause the enzymes to stop doing its job or catalyzing the reaction. All right, so here are two other possible regulators, AMP and PEP. AMP is produced when the cell breaks down a lot of its ATP, and the cell breaks down a lot of ADP into AMP. AMP is often referred to as the cell's starvation signal. It's like, I'm hungry. AMP is, I'm hungry, I'm hungry. Would a high level of AMP activate phosphofructokinase, or would a high level of AMP inhibit phosphofructokinase? Well, a high level of AMP, which happens after the cell has used up a lot of its ATP, should signal the cell to speed up phosphofructokinase, thus speeding up the breakdown of sugar and the production of ATP. And so AMP is a phosphofructokinase activator. All right, PEP is produced at the end of step 9 in glycolysis. Explain what the cell should logically do in the presence of high PEP levels. So if there's a lot of PEP, that means there's already a lot of sugar being broken down and a lot of ATP being produced. If there's a lot of PEP in the cell, glycolysis should be slowed down. PEP is a phosphofructokinase inhibitor. That's a mouthful. Maybe it should be PFK inhibitor. Either way, there you go. So you guys had these. We didn't have different color paper necessarily. Maybe you did. But you should have had step 1 paired with the hexokinase and glucose. Then step 2 should have been the glucose 6-phosphate isomerase and the glucose 6-phosphate G6P. That's the step 2. So make sure, double check that yours are in the right order. Step 3 is the phosphofructokinase and the F6P. Then we have aldolase. And remember, I'm not going to have you memorizing the term or the actual enzymes and names like glyceraldehyde 3-phosphate and hydroxyacetone phosphate. I'm not going to have you memorize those names, but what you need to understand is the overall cycle and how it works and the things that are happening. You will need to know the products and the reactants of each cycle, maybe just not the intermediate steps, okay? So here's the next step. Step 6 and step 7. Make sure yours match. It can be, I don't know that it mattered where you pasted each thing. I can't remember if it was labeled. No, so it wouldn't have mattered. Just as long as you are putting the steps in order with their correct molecule and enzyme. Okay? All right. And lastly, oh, my baby's back. So lastly, you're on the last page with the enzymes and substrate. Sorry, didn't mean to zoom by it. Okay, and your final product is 2 pyruvate molecules. So this is all of your work for the last week. We'll go over the other pages after I grade them.